Hello, Dr. Jeffery. You are a professor of neuroscience at University College London. So welcome to Modern Healthspan, and thank you for coming on our channel today.
Oh, it's my pleasure.
So, Dr. Jeffery, so you've been studying photobiomodulation, among many other things, and some fascinating effects of light on biological systems, of which humans are one. So, how does light affect biological systems? I mean, you think about the eye, right? But even if it shines on the skin, it has impact. So, at a high level, I mean, how does it do that?
Well, if you think about light in a certain way, light provides energy. Okay, it puts energy into a system. That's the first way to think about it. And for millions of years, or in actual fact, billions of years, the driver for evolution, where evolution has obtained its energy, is from solar light, sunlight. So our bodies, you think about us as the apex primate. We've evolved in Africa naked for 5 million years. We've had a 12-12 light cycle. We've had great sunlight in that environment, and that sunlight has been absorbed as it passes through our body. Sunlight will actually, you can measure sunlight coming out the other side at very long wavelengths. And when it passes through your body, it deposits energy. That energy is used by the body. And the particular thing that I'm interested in is something called mitochondria, which are terribly important. They run your metabolism. They provide you with the energy to get up in the morning. They do so many things. Critically, when you undermine your mitochondria have the ability to say it's time to die, and they can kill you. Keeping them happy is a really important thing in life, and sunlight has a very strong or light of certain wavelengths has a very strong relationship with mitochondria.
Okay, so if we looked at the mitochondria specifically, so and which is the thing that you study mostly, so how does light affect mitochondria? So, I mean, I think it increases the amount of ATP they produce, which is the energy. So how does it do that?
Okay, so there's something called a respiratory chain. And if we think about this as a series of railway stations, so a train goes along the railway stations, and when it gets to its final stop, that is the production of ATP, which is the energy that the body uses. Various parts of that chain, various railway stations on the road specifically will absorb that light. In absorbing that light, they increase the efficiency of the mitochondrial chain. Now, as you get older, you can think about it, as you get older, the railway tracks on which the train is running become distorted, the train slows down, things just aren't working terribly well. Anyone who travels on trains in the UK will know all about that. But it's simply, it is not complex. We watch light go in, we watch what light does, and it increases the efficiency of the train going along the railway line. Now, we know quite a bit now about which bits are absorbing, how they absorb. In my lab, we can actually physically watch them do that, because as they absorb light and as you get them to work, they change color. So if you've got a really quite expensive piece of equipment, basically an expensive spectrometer, you can watch a mitochondria and you can watch what it's doing and how busy it is, and it changes color. So you could imagine maybe you and I running up the stairs. As we run up the stairs at home, by the time we get to the top, we're vasodilated, we've changed color. Mitochondria do exactly the same. And for years, we haven't paid attention to this because we've got a metric that tells us, first of all, how healthy our mitochondria are and how hard they're working. So the whole issue of photobiomodulation stayed in the cupboard for a long period of time because we didn't have a regular handle on the mechanism. I think we've got enough of a handle on the mechanism now to make exploration in this area plausible and worthwhile, and with a very, very clear clinical and health span benefit at the end of that.
So I was going to ask this question later, but if so I've got light coming in and it's changing the protein in the mitochondria. Does it have to be at like a specific wavelength to be able to like excite electrons or whatever to make that change? Or will anything, or how wide can the wavelength range be?
I think we're pinning that one down quite well. It's the longer wavelengths of light. So we will say red into infrared. In terms of numbers, it's about 660 nanometers, which you would see as quite deep red, through to 900 nanometers, which you can't see. But once you've gone beyond about 750, 720 nanometers, you can't see the light anymore. If you turn the energy up a lot, you can just see a dim red light. But those are the wavelengths that are important. I think there are a few hotspots in those wavelengths. However, anything in those wavelengths will have impact. Our solar environment, solar light, is particularly rich in those wavelengths, but we ignore them because we can't see them. Our visual system can't see them. And that you tend to ignore what you can't see. And that has been a bit of an issue. So yes, it's sitting under, I don't know, a fluorescent tube is going to do nothing for you. Going outside, you're going to get a lot of infrared light. And if you go back to our old incandescent light bulbs again, you'll get lots of infrared light.
Right. Okay. And kind of anything in that area can have this positive effect.
As far as we know, yes, it can. As I say, I think there are a few hotspots, but the trouble is you could waste half your life going nanometer by nanometer by nanometer, trying to find out exactly what is the hottest spot there. I think if you think about evolution, we've evolved under solar light, which has got this very, very broad spectrum. The infrared in solar light goes up to about 2,500 nanometers. That's probably not much good to us, because when you go over 900 nanometers, most of that light will be absorbed by water, not by the biology in your body. So what we're looking at is really 650, 660 through to 900 max. In the past, everyone has used 670 nanometer light. Now, there is no reason for that. The only reason is the first people that did the experiments, started on these experiments, which were really a great 2 or 3 groups in Australia, happened to have a device which produced 670 nanometers. So that became a benchmark. So we know a lot about what 670 nanometers does, but you can do almost exactly the same with 850 nanometers, which you can't see. So I'm not going to pull that spectral range apart. It's too much work for very, very little reward. Let's just make sure we get a broad spectrum of infrared light in our lives.
Right. So if we took if we look at the whole light spectrum, what does, I mean, because I guess it goes from ultraviolet all the way through to infrared, and the sun produces all of these, right?
Yes.
So where does our spectrum fit in? The one you're interested in fits in at the long wavelengths, right?
Yeah. So yes, you're absolutely right. Solar light goes from the just over 300 all the way to 2,500. The key thing in solar light is it has a natural balance between blue and red. Okay, I'm using blue and red as a very, very loose terms here, right? But if you looked at solar light, there's a balance in it. And that balance, I think, is the critical thing. Now, it is definitely the case that we have ultraviolet, and we know ultraviolet is important because it produces vitamin D, and we have a vitamin D crisis in the Western world because we're not getting enough sunlight. But we've got a rather a condensed range in the ultraviolet through to about 400. But we have this enormous tail that goes out a long distance in the infrared. So I'm not sure where you actually technically call infrared starting, but you could actually say, let's say it's what you can't see, 750 to 2,500 nanometers. There's a lot of it out there. So that balance between blue and red has always been there. We've evolved with that balance. If you upset that balance, things can go wrong. And I think the other thing to also remember is the ultraviolet. It doesn't really penetrate your body very much. It's why you get sunburn, because all the energy is absorbed at the surface. Whereas the infrared, I mean, we measure it coming through your body. We've stood people in sunlight and measured the light that comes through their body. And when you go above 780, 790 nanometers, you can measure it coming through. Not all of it, only some of it. So that means the red's actually potentially a lot more important, because it's getting to your liver, it's getting to your lungs, it's getting to your brain.
Right. So if we went back to, okay, the 670, let's take 670. So how deep does 670 go? Yeah, I mean, can you get to your liver?
Oh yeah, it certainly can get to your liver. It won't come out the other side. It's not quite long enough to do that. But 670 will certainly go through your skull. So for instance, in looking at aging, we have used 670 on the eye to improve vision in people that are older people, very successfully. And it transpires that when we do that, we're also illuminating the inner ear. So, it is going through, longer wavelengths go through bone. You automatically think it's bone, that's going to stop it. No, it doesn't. Longer wavelengths will go through bone, and you can barely see the bone as a very, very distant shadow. But the 670 will not go all the way through the body. Right. But it will certainly go pretty deep. I mean, I strongly suspect, in fact, we've argued this before, that because your chest is vacuous, it's largely an empty space, we can use 670 to get to the heart. So one of the arguments that I think has a lot of traction is in terms of things like if you have a heart attack and your cardiac tissue becomes ischemic, right, doesn't have enough oxygen, we know that 670 nanometer light improves the ischemic situation. So I would love to see a 670 light actually in an A&E unit. And when people come in with an ischemic crisis, I mean, we know from all the science that the red light, and 670 in particular, can help ischemia. It costs nothing. It's going to do you absolutely no harm. And if it saves 1 in 1,000 people, that's a winner, as far as I'm concerned. Yes. So, yeah, different wavelengths, longer the wavelength, the greater the penetration.
Okay, if we think about dosing, so it's like power and time, or are there other parameters? How do you, when you're doing an experiment, how do you I guess calculate what dose you will use?
That's a terribly good question, and it's also an extremely demanding question, because with the red light research, we have too many variables, so we can't isolate one variable very easily on its own. You have wavelength, you have energy, you have time, and you have time of day, because your response to red light changes at different times of the day. So how do we manage that? Well, I don't have access to enough subjects to pull that one apart. We've done a lot of experiments on animals, and what you find in this is highly conserved in evolution. What you find in a fly, a bumblebee, or a human is exactly the same in terms of red light metrics. So we know that if you go below around 1 minute, 1 minute 15 seconds is the break point. if you give red light at any energy below that time period, nothing will happen. And it's not like aspirin. Take 5 aspirin and your headache will go away quicker than if you take 1 aspirin. It is something that we now know is a switch. When you do something, you turn the switch that improves mitochondrial function, and that switch, once you've turned it, will last 5 days. Again, doesn't matter if you're a fly, a human, whatever. Now, physiologists find this very difficult because in physiology, there aren't a lot of switches. There are a lot of dose relationships. So when we've done work on aging people and aging eyes, and we've managed to improve vision, we say to people 3 minutes in the morning. We know 3 minutes is an overkill completely. But then if you've got someone that tends to chisel things away, so you say to them, oh, it's 1 minute and 15 seconds, someone's going to do a minute. Someone's going to chisel it away. And so for that reason, we've gone over the top and we say, do it for 3 minutes. Now, when this scenario came out, people had a fixed energy for using this, and it was 40 milliwatts per centimeter squared. Bit bright. So what we did do is we started reducing that and still looking for an effect. And so we had a project in Moorfields Eye Hospital where we took some people and we just reduced the energy. And so we can now deliver an effective dose. Again, remember, it's a switch at a much lower energy and get as good a physiological effect as if we give high energy. So we can now deliver something that is very comfortable. And no one's going to, we've got clinical trials with young children. We've got trials with older people, and they tolerate it very, very easily. We're down to about 5 milliwatts per centimeter squared, which is, if you don't know what that looks like, it's a relatively dim bicycle light.
Right.
But it's not a bicycle light because the wavelength's longer. But that's what we're really talking about. We're talking about something which is very comfortable.
Interesting. Now, but this is for mitochondria. This is for helping mitochondria, right?
Yeah.
I mean, because people use red lights for inflammation and many other things, right? Which we won't talk about, but that may have a different dose response.
It may do. Now, red light for inflammation, we think has been, well, we know it's been used for ages. Vets use it very regularly on racehorses. I spent a lot of time talking to vets at one point. If you've got a very expensive racehorse, you want to look after its muscles. If it's got muscle strain, then you really want to pay attention to it. I strongly suspect the metrics are very similar, because what runs inflammation is mitochondria. Mitochondria have a very, very strong influence over inflammation. So if we take an old animal, let's say an old mouse, the eye burns more energy than any other part of your body. It ages faster than any other part of your body, and you start to get systemic inflammation in the back of the retina. When we give red light, which affects the mitochondria in the retina, inflammation goes down. It's very clear. Lots of separate metrics we've used. The mitochondria, when they're unhappy, drive inflammation. When ATP production goes down, something called reactive oxygen species, ROS, goes up. Now, ROS, we can think about ROS as, I think about mitochondria as a battery. When the battery is running well, it produces ATP. When the battery is not running very well, it produces more ROS, and that's like a battery leaking acid. And when it does that, inflammation starts to be driven. So we know red light reduces ROS, increases ATP, and when we look at a whole range of inflammatory markers, they go down. Now, some go down a lot, some go down a little, but There is a fundamental relationship between light and mitochondria, and it doesn't matter what animal model you do it in, and I don't think it makes that much difference which tissue you use. If you use a tissue that doesn't have much in the way of mitochondria in it, a tendon, the effect is much more reduced.
Right. Interesting. How did these people do it every day? You said the effect lasted 5 days. When they were doing the eye, is it every day?
Yeah, it lasts 5 days no matter what you do. We are at 5 days is a very, very difficult kind of concept to get over. If you're saying to an elderly person, do this every 5 days, they lose track. They lose track. Now, was it a Monday when I did that last, or was it a Tuesday? And is that 5 days or 4 days or 3 days? We tend to say to people, just do it daily. Build it into your routine. There is absolutely no harm in doing it daily. But if you sit there and you do it daily and you sit in front of, your red light for an hour, the whole thing falls apart. It's relatively ineffective. So you go past some point of efficiency. The best thing, without any doubt, is 3 minutes. Build it into your life. Do it when you're making your coffee in the morning, when you're listening to the news, which people seem to do very, very easily. Do it daily if you can't remember what 5 days is. And look, I'm not young. I don't remember what 5 days is. So when I come into my office, I cycle to work, I get into work, I start changing out of my cycle gear, and I just turn a big red light on that bounces light off a wall. And that is what I do. And there are a few days when I miss it for one reason or another, but I probably do it 3 days a week, And if you miss it, if you miss something, it's not the end of the world. It's just got to be a bit of a pattern you build into your life.
In the connection between nutrition and optimal hormone function, one mineral stands out. That is magnesium. It regulates hormones like insulin, thyroid hormone, estrogen, progesterone, testosterone, and cortisol. For example, a study with 399 men over 65 found that higher magnesium levels were linked to higher testosterone levels. Magnesium breakthrough from Bioptimizers is the only supplement that offers all 7 key forms of magnesium. This includes highly bioavailable forms like magnesium glycinate, malate, and orotate that raise cellular magnesium, supporting optimal hormone production and regulation. Since I started taking Magnesium Breakthrough, I feel more energized, my mood is more stable, and I've noticed improvements in other areas affected by hormone imbalances, such as sleep quality. If you want to try Magnesium Breakthrough, just go to bioptimizers.com/modern and enjoy 10% off with the code MODERN10. It comes with a 365-day money-back guarantee. And if you subscribe, you will ensure your monthly supply. Thank you for your support. Be good to talk about some of the animal trials you've done, and what benefits you've seen. So, could you talk? I believe you've seen some kind of life extension and Healthspan.
Very definitely. So we feel our way forward in science. There's no point in jumping into some big clinical trial if you're not sure where you're treading. So what we do is, because we know this mechanism is highly conserved across species, we started off with working on fruit flies, which is what a lot of scientists do. And we started working on bees. Now, there's a good reason for working on bees, because insecticides that people are using, some of the vicious ones, work by destroying mitochondria. So we had a sideline project where we were giving bees red light as well as giving them pesticide, which was very, very successful. But the key point is that the total lifespan of the animal did not increase. Okay, so let's say a bee lives for 70 days, a worker bee. What did happen, which was very dramatic, was the average life expectancy increased very significantly. Because if you look at a big population, as you start to reach your middle age period, you start to find you've got an increasing death toll. The death toll in the middle-aged bees and the flies reduced very significantly. So by the time we got 3/4 of the way through the natural lifespan of the animal, we had many more animals alive when they were having bursts of red light. And those animals had a better health span because we asked them to do things. We asked them to climb up walls, and they could do that far more efficiently. That made sense to us because muscles have got a lot of mitochondria in them. So no overall extension in absolute lifespan, but a very significant extension in average lifespan. If we think about ourselves, you could say, well, you know, a large cohort of those people dying in their 50s and 60s, we could drag those through and they'd be walking down the road better than without the light. That's an assumption based on the animal work. But I have found absolutely nothing that contradicts that. And those experiments have been repeated time after time after time. And they're a lot cheaper to do than doing on humans. And also, actually, when it comes to the bees, that was fun.
Yeah. Bees are a bit more charismatic than flies, I have to say.
Exactly. And I would say that, you know, when it came to the bees, I don't have commercial interests of any kind. There are commercial beehives in France now that have red lights in them. And the hives are doing very well. And in America, because they've used far too much pesticides, they've sanitized big areas of the countryside, so there are no pollinators. This is particularly true of the almond harvest in California. So they truck thousands of beehives up from Texas and Florida into California. And of course, many of those beehives die through stress. Red lights are now going into those trucks to see if they can keep those bees alive to reduce the stress of being transported around to different places. And the moment you transport a bee to a different place and you open the door, the bee comes out and goes, "Where am I?" and gets really quite stressed by the whole thing. It likes to have its patterns, its hive in one place. So that was interesting. That was fun.
Yes. Have you heard of the ITP, the Interventions Testing Program?
No, I haven't.
Okay.
So who runs that?
It's three labs. It's the NIH, the National Institute in America. And it's 3 labs that they run tests on mice to see if there's any intervention which will extend their lifespan.
Right. Okay.
And normally it's things like rapamycin, right?
Yes.
I just wondered whether you could give them red light.
We were going to translate the experiments from bees and flies into mice. The trouble is, if I buy a mouse, it's ridiculously expensive. It's like £50. You know, it's very expensive. Then for me to keep it is about 40 pence a day. Now, if you're thinking about a big cohort of experimental animals and a big cohort of their controls, that's just too expensive. So what we have done is we've made that jump very often from insects through directly into humans. We have had a few stopping points where we thought, hang on, we just need a little bit more security here. Let's run a few things on the mice, but lifespan and health span, it would just be phenomenally expensive.
Yeah,
I would say that if we do take old mice and we do give them regular exposures to long wavelength light, all the indications are very clear their health spans are getting better. All of, you know, so I strongly suspect that experiment will be done by someone. It won't be done by me because I don't have the money, but I think the outcome is relatively predictable.
Interesting. Okay. Yeah. No, I think, but anyway, if we could persuade the ITP to try it, but then you wouldn't have to pay for it. So, but one other thing that I did see that you saw improved memory. So also improved cognition.
Yeah.
Could you talk about that?
Yeah, certainly. So if you think about any cellular system, where's most energy being used? Most energy is being used. So let's think about the nervous system. It's being used to keep a membrane pump going. Okay, so it's pumping ions in one way, it's pumping ions out the other way, so that cells have got a membrane potential. This is a big, big absorber of energy. So in the brain, your membrane potentials on your cells go down with age, and your mitochondria suffer. If the mitochondria are not providing the ATP, then you can't keep your membrane potentials in shape. So we had a super great graduate student, and he said, look, if we're doing what we say we're doing, then we've got to be affecting the whole of the nervous system. Let's take some old flies and let's give them some memory tasks. And I kind of thought, oh, crikey, you know, this is going to be a difficult one, but he was very smart. And so he taught flies to run down a tube, because they'll always run towards a light, and they run towards the light. Then he did something horrible to them. He took some filter paper and he soaked it in quinine and put it halfway down the tube. Flies hate quinine. They loathe it. So the old flies would run down, they'd get to the quinine and they'd go, didn't like it. The young flies remembered that the following day. They remembered there was quinine and they didn't go down the tube. The older flies, their memory was much poorer. They kept making the same mistake. If we took those flies out and we gave them a burst of red light, I think we did it over 2 or 3 days, their memory was much greater, much better. Many more of them didn't go down the tube because they remembered there was a toxin down there. Now, we do similar things with them in terms of we put them in strange environments. And if you put a fly in a strange environment, it'll go around it in a very, very stereotypical way. It looks for borders, it looks for edges. Older flies don't do that. They just stumble around. Again, when we gave those after testing, and then we gave the same older flies exposure to long wavelength light, bang, they were better. We take those flies up and then we extract their brains and grind them up. They had far more ATP in them, and their mitochondria were far healthier. So there was a direct link between light, what was happening to their mitochondria in terms of how much energy they were producing, and behavior, which is the critical thing. It's the behavior that's the critical thing. So yes, now I have not done experiments on mice in this area, but there are a number of very good labs that have looked at mouse models of dementia, and they've got improvements with red light. Now, why these labs have not made the transition to moving into a clinical trial, I don't know. Many labs are bound by the methods that they've got on their shelves. They do what they can do. So, you know, you say to people, hang on, look, you've got all this absolutely great data, and these are the really great Australian labs. You've got all this great data. Why don't you translate it? And it has, I don't know of any of them that has translated it, but they all go, yeah, that'll work. They don't know, they haven't proved it. But the great thing about this whole area is, what's the downside? It's totally safe. We've got ethical approval time after time after time for old people, even for very young kids that have got a genetic problem with their mitochondria. It's worked every time. It's perfectly safe. What's the worst thing that could happen? The worst thing that could happen is in some situations it wouldn't work, and then there's egg on the face of the experimenters who actually proposed it. But there's no harm in it. And in actual fact, it doesn't have to be expensive. It can be incredibly cheap. You know, I say to people, if they're going to go buy devices, I say, well, okay, maybe that device, I think that device is safe. I have no commercial interest. And I say, why don't you share it with your friends? You know, there's no reason why you can't share this with your friends. Pass it around. You know, you've only got to do it a couple of times a week. Pass it around. So yeah. I think we have got interest from nursing homes. And I'm trying very hard to put together devices that can just be incorporated into walls. You don't even see them. And we know that we can do that with long wavelengths of light that don't disturb your vision, 850. You don't even see 850, you just feel a bit of warmth in the distance, and has the same effect. Why are we not putting those in nursing homes? You know,
Mm.
that's a big push for my lab, and that's why I'm talking really hard to lighting engineers and to architects. Let's get these devices in. Yeah.
Right. So I think you kind of said this, but just to confirm, like, so there's no, you've not seen any side effects or any contraindications from using red light?
No, we've got some people who, I have two aged aunts who have been having red light now 6 years, 7 years. No contraindication whatsoever. There's one thing that makes me nervous. I mean, it's cheap, it's very effective. And that is that if you expose some cells to red light, some cells that naturally proliferate, let's say even just bacteria, they proliferate a little faster. So we've all got together as a community and we said, one thing we will not do it on is we will not use red light on, say, for instance, patients with cancer. I have no evidence whatsoever that it's harmful, but just putting two and two together, there is a possibility that it might be detrimental in that situation. And so on all our ethics applications, we put down, we will not use this on people with proliferative diseases. I have absolutely no idea whether it's a problem or not, but let's be careful. Yes. Let's be careful. Yeah.
Right. Okay. So turning to, so you have done some human trials, which I found really interesting. Especially and one of them that there's probably a lot of interest to our audience is the lowering of the blood sugar. And so could you talk about the way that works and also the fact that it was systemic?
Oh yeah, I know that was I was driving 3-hour journey to a research lab somewhere at a different part of England. And that's my time for sitting and talking to people and just thrashing out ideas because no one disturbs you. And a very good colleague of mine, Mike Powner, were driving along and said, well, you know, if we're making these mitochondria work harder, they've got to use more fuel themselves. And what fuel do they use? What do they burn? They burn carbohydrates, sugars, and they burn oxygen. So I said, we were driving along, I was saying, oh, that would be too good to be true. And he said, but it has to be true. You have to burn more carbohydrate. And the only place you're really going to get fast carbohydrate is from your blood sugar. So we did an experiment on bumblebees. And we made bumblebees diabetic, and that worked brilliantly well. And we got ethical permission for it. And then we took people and we gave them a standard glucose tolerance test where you drink a disgusting amount of glucose and then you prick your finger on a regular basis and you test your blood glucose levels. And then we tested how much oxygen they were consuming by putting a tube up their nose and measuring their CO2 production. So everybody, after you've taken a big load of glucose, between half an hour and an hour, you get a big peak in your serum glucose. And the controls did that. The experimental people that we gave a burst of red light to, and we gave this a burst of red light on actually a relatively small region of their back, their blood glucose levels didn't peak in the same way. And the reduction was very significant. The reduction was so significant, I said to my colleague, Mike, do it again. Do the whole thing again. I'm just not happy about this because, to be honest, if we get this wrong, we'll be cut to pieces. And we did it again. We got exactly the same effect. Now, these were healthy individuals, right? These were relatively healthy individuals. We are now 3/4 of the way through a project where we're doing it with people with type 2 diabetes. And so far, we're getting exactly the same result. Now, why? So that makes sense. Mitochondria improved. That means they need more glucose. They need more oxygen. Why does this work with just a little area of illumination on your back? Well, the thing that's out there that we had paid relatively little attention to was that the moment you disturb mitochondria in one part of the body, they signal to mitochondria across the body, and they do that well within 24 hours. We know there's something going through your body called cytokines. They're inflammatory regulators, and you can buy a cytokine kit from a company, and you can just put body samples on that, and it can measure your cytokines. And we've done that. We did that originally with mice, and a burst of red light changes all the cytokines. So that means your message goes around the body. It may not be the key message, but it's certainly a message. You go back 15, 20 years, people were doing experiments on little worms, changing mitochondria in one part of the body and noticing that all the other mitochondria in other parts of the body changed as well. So there is some way in which these mitochondria are talking to one another. That and there's a diabetic lab, a very good diabetic lab in the US, and they had a very similar result with affecting what goes on in the retina by shining light on the back of an animal. And the guys in Australia did exactly the same thing. So that made me feel good. It made me feel good that other people were at least replicating the paradigm. So my dream, which I said to the lab many years ago, as I said, I want to be able to have a system where you go to a cash machine, okay, and it takes you 1 minute, 15 seconds to get your cash out. I'm going to delay you in some small way. And you put your hand down on the cash machine, and that gets, you have to put your hand down, maybe to identify yourself, right, your fingerprint. And when you do that, you're identified by an infrared light. And we all said, yeah, Star Trek. It's not Star Trek now. That is theoretically perfectly possible, because I do not have to deliver the light to the part of the body that I necessarily want to treat. Also, we know that deep red light, say at 850 nanometers, is going to go straight through your hand and be absorbed through the full depth of your hand. This is potentially doable, and you can use at the same time that handprint to identify as a secondary identifier as you as a person. It may not come out that way, but in theory, we don't have too much of a problem with progressing down that pathway.
And so the light is before the glucose or after you take the glucose test?
No, we gave the light before people had the glucose load, right? So you could call it prophylactic treatment. We treated before there was a problem. Now, I don't know how the situation would change if I gave you the glucose load and then at fixed time points afterwards fired up your mitochondria with red light. I don't know that. I mean, first of all, from a big point of view, does it matter? You know, we know we've got a way of doing this. We also know that in big population study in the Netherlands and in the UK, people with type 2 diabetes have lower peaks in their blood sugars if they work outside a lot, where they're being exposed to a lot of long wavelength light, compared with office workers who are not being exposed to long wavelength light. So the whole envelope here is looking relatively watertight. You know, in principle, this works. Am I particularly concerned about when you get the burst of red light? No, not really. I always prefer, if we can, to be prophylactic, to treat you before there's a crisis. But I'm not sure it's particularly important.
Right. So like before I go and have a big carb load, I could shine some red light on myself and it would lower the spike.
It would lower the spike. Now, we also now have people walking around with, instead of pricking people's fingers all the time, which it's very difficult to get subjects for that. I mean, it was a nightmare getting subjects for it. Yeah. You know, we now have constant glucose monitors, which we put on the arm, and we're now getting a profile over a number of days in these type 2 diabetics, and it's very clear. Your blood glucose levels do go up, but nowhere near as much. And when it comes to treating diabetes, someone can have high blood glucose. That's not the issue. The issue is the spiking. How much are they spiking and how sharp are those spikes? That's what your blood vessels really don't like.
Right. Good. Yeah, interesting. And that, so what happens? I just think about this. So what happens to the extra ATP? So we're burning all this glucose, we're turning it into ATP. But I mean, doesn't the cell have a mechanism that says, okay, I've got enough ATP and I turn it down?
You know, that you'd think that when you look at me, you'd say, you know that, don't you? And funnily enough, we got through 5 years of research without us asking that question. And that was a big failing of us. That was a terribly big, and it took an outsider to come in and actually say, what's happening to all this ATP? Well, the first thing is that if you do generate lots of ATP, you'll start storing it as ADP. So there is a storage mechanism there. But the other thing is that you are increasing metabolism. So it's something we haven't explored as well as we should do, but it's on the target list. You're not only producing more, but you're burning more. So that any one particular point in time, and this is a problem, ATP is quite hard to measure. And we can't measure it in people in a constant kind of flow. But we think that what you're doing is you're generating more ATP, but your whole body's using more ATP. And there is some evidence that people with certain patterns of red light exposure have slightly higher body temperatures. Now, mitochondria themselves are super interesting because they run at high temperatures. Now, we don't know exactly what that temperature is, but we're thinking that I mean, there's certainly agreement that it's in the 40s, the low 40s Celsius. So it's way above 37.5, your body temperature. And it's one of the reasons your body warmth is coming from mitochondrial action, right? A lot of it comes directly from mitochondria. So it's a super good question because the body has to have a way of controlling it. I think we're just turning up metabolism. It's relative, and some of it we're throwing into ADP, which is a way of storing it. There is, as I say, there was a great experiment done on athletes being given red light. Now, they're not good people to give red light to because really want to do this to older people. But Oxford Brookes University did some great experiments getting them to run and testing all their bodily functions. And there was some indication those people were losing weight. Wasn't significant, but I think that was a move in the right direction. And that's where I've gone back to do an experiment with mice, and we're actually sort of measuring the weight in mice. If we give them lots of blue light that runs their mitochondria down, mice get very fat. And they get very fat actually surprisingly quickly. So, and their blood glucose levels all get disturbed. So there's got to be a link there, but I'm not quite sure what the link is. That is a question. What are we doing with all this excess ATP? That needs to be hammered as a question.
Right, but at the moment it seems to go into the metabolism.
Yeah.
So, have you ever looked at cognition in humans with red light?
No, I haven't. There are a lot of experiments going on with red light and cognition. My problem is I know data that comes out of certain labs. I know those labs, and I think their data's always good. Their data's always reliable. One issue in science is very often people will move into a field like the red light field. You don't know who they are. You don't know about their data. You read their papers and you're going, oh, your numbers are a bit low and I don't know anything, and there's a lot of stuff being published that we cannot completely rely on yet. So it's a very, very obvious question because we do know that in aging, in the brain, ATP goes down, mitochondrial function goes down. If you've got less ATP in the brain, you've got less energy for keeping those membrane potentials pumped up as we, as we know, that absorbs enormous amounts of energy. It's pretty obvious. It's pretty obvious. And those wavelengths of light will get into the cortex. If we take 670, that'll go through the bone. It will get into the cortex of the brain. If we use longer wavelengths of light, it will go right down to the deep levels of the brain. And again, remember, it's cheap. We've got no obvious downsides to it. I must say that if I thought my cognitive abilities were suffering badly, as a scientist, I'd be making sure that I did get a burst of light in the mornings. Yeah.
Right. So you did experiment on eyes and particular macular degeneration. We kind of touched on that. Did you? Yeah. Can you talk briefly about what you saw in terms of?
So macular degeneration has got a strong mitochondrial component. It's a really massive disease in terms of people are if everyone lives to the age of 120, 50% of them will have macular degeneration, and they'll be blind in their central retina. So it was very obvious. We kicked an experiment off, we did it, and it failed. We went back and we looked at it, and we found out why we think it failed. We selected the wrong subject population. Disease was too well established in them. The controls were their partners, because they live in the same house, they have the same food, they're similar ages. Those that we gave red light to did stunningly well, really stunningly well. However, there is a tighter experiment that has been done, much tighter experiment on macular degeneration. I know the person that has done that experiment very well, and the data are very positive. Now, what they did was they grabbed their patient cohort when when the first signs of disease were present, which was, in this case, it was drusen, these deposits that form in the eye. Some of our patients already had that central part of the retina starting to degenerate. Once the degeneration had set in, it was much harder to deal with it. The outcomes of that are great, and I actually rang the guy up and I said, this is fantastic. You've done everything, you've corrected all the mistakes that I made, and it was great to see the result. Really good to see it. So if we've got the other great data sets that come out are things like Parkinson's disease. So if we look at a cohort of us as we age, macular degeneration, diabetes, Parkinson's disease. Parkinson's disease is fundamentally a mitochondrial disease. If we catch these things early, we can probably deal with them in many people and improve the quality of life.
That would be amazing, given that I'm more familiar with Alzheimer's, but it's like, given that there's no other treatment for them at the moment.
Yeah. The problem with things like Alzheimer's is you really do need to run that trial over a long period of time. I don't want to really know what happens to you in 2 months. I want to know what happens to you over a 2-year period. That makes them expensive, very expensive. If we had a big donor here, that would be exactly where I'd be jumping. But the Medical Research Council are not going to pay for that, or unlikely to pay for it.
Right. So blue light. So we talked about red, blue light. So is it all bad? And because you talked about the way that it, I guess it deactivates my mitochondria and makes you fat.
Well, it runs the battery down. We know that. It runs the battery down. We know that LED lighting is very strong in blue and no red in it at all. And when you're sitting in a place that's very hot, nearly all your glass will be infrared blocking. Okay, right. So you're getting lots of blue light from your lighting source. You're getting no red light from it, and you're getting no long wavelength light in through the window. LEDs are a problem. They really are. They came in the the 1990s. They suddenly were so much more efficient than old incandescent light bulbs, but there is a price to pay for this. And we need to think about that one very hard, how we get around it. The architects and the lighting engineers are very much on board now, because there's a litigation point at this also. If you're sitting in your office and you start developing type 2 diabetes, you're going to be very annoyed with your employer if they put you in a building where there's no red light. Now, at the moment, the knowledge base really isn't there for that, but the knowledge base will be there. It will be there in about 3 or 4 years' time. And of course, loads of people are becoming pre-diabetic and loads of people are going to start complaining about it, not surprisingly. So if we look at the astronauts on the International Space Station, they've got all harsh LEDs. They're up there for months on end. Many of them are becoming pre-diabetic. Okay, all of their glass is infrared blocking, and all of the glass faces the Earth. It doesn't face the sun anyway. So there's loads of data to say that we need to think about LEDs. They were fantastic. They've saved us loads of money. We need to go back to another form of lighting. The downside of that lighting is it will generate heat, which we don't want. We've got to find a way around that. I'm certainly talking to the glass industry and to the lighting industry about it, and they're not hiding from the problem.
Interesting.
Yeah.
So do you think blue light blocking glasses help at all? Do I need like a blue light blocking suit?
No, because you need blue light anyway, because you need to synthesize vitamin D. Okay, so this is all about developing a healthy balance in your attitude to light. It's not about saying this is all good and this is all bad. It's about a balance. I must admit, I had the whole of the lab walking around with yellow glasses for a couple of weeks, and it's one of those experiments that everybody was very interested in because all the other labs could see my guys walking around with yellow glasses. It had no effect whatsoever. Now, I still think that there's something in that, but I got the question wrong. I need to ask the question in a different way. And I don't know what that is. Yeah. Science wouldn't be fun if you answer all the questions. You'd still need the problems there.
Right. Well, I mean, hopefully they help you sleep better. But I mean,
I think that is true. That
Yeah.
is, I don't have a sleep problem, but I've heard people say, yes, blocking that blue light late in the day does help sleep. And that's great. But it's not doing what I want it to do. Never mind.
Yes. Okay. Okay. So if we thought about takeaways from all your research. So what can people do now to reduce the impact, or perhaps to benefit from red light? Do you have any like suggestions?
Oh, definitely. Go for a walk in the park.
Right.
Okay. Go for a walk in the park. So here's the other thing. When you hold a leaf in your hands on a hot day, it's not hot. It's cool. Why is it cool? Because it reflects all the infrared light back. Doesn't want it. That means your walk in the park is a walk in an infrared-rich environment. Go back to a situation where for millions of years you've evolved into a natural lighting situation. If we're not going to do that, then think about your light bulbs. Think about old incandescent light bulbs. You can still get them. You get them on Amazon from China. Think about, you don't have to do anything drastic. You know, just change your lighting, walk outside more. I mean, particularly think about it in winter in the Northern Hemisphere, where, you know, many people will get very, very limited light exposure. That's all you have to do. You have to move back to a situation which is closer to where you've evolved for millions of years. You can very easily go and buy red lights. Many of them are on Amazon. Many of them are a bit, I have very mixed feelings about their quality. But why don't you just go for a walk? Why don't you say, I'm going to go out into daylight, and you think, I'm not going out into daylight because it's cloudy today. All the infrared light goes straight through clouds. It doesn't do anything, right? Scatters it around a little bit. Just go for a walk. If you go for a walk every day for an hour, and I think as we're aging, that's important on loads of metrics. We're using our muscles. You know, when we're walking around there, not only are we getting red light helping our mitochondria, but we're burning our blood glucose because we're going for a walk. You have a massive glucose load, put a patch on and go for a long walk. Your blood glucose won't peak in the same way because you're burning that energy in your muscles. Lots of really simple things. I don't believe, in terms of healthy aging, I don't believe there is a route that needs to be taken that involves you spending vast amounts of money at all.
Right. Yes.
I hope so anyway.
Yeah. No. Yeah. That is certainly the goal. Yeah.
Yeah,
I
yeah.
mean, is there a best time of day for getting red light, going out, or
Yeah,
however you do?
definitely. Doesn't work anywhere near as well in the afternoon. If you think about it in terms of evolution, when you wake up, let's say you wake up in your cave, you're very vulnerable. You're super vulnerable. You have to wake up and you have to be on the ball. That's why when you wake up in the mornings, your blood sugars peak. You have a whole series of hormones that peak in the morning, and your ATP is starting to peak. When your ATP is moving and it's going up, that's when you can push it and accelerate it. If you start using red light late in the afternoon or night, it has very, very little effect. So if you go and book a session using red light in some therapy or health center somewhere, don't book it in the afternoon. Okay, it's got to be something you should really do in the morning. The body's actually saying to you, wake up, you're vulnerable, get out into sunlight, move your ATP up to match the changes that are happening in your blood glucose and the hormonal changes that you go through during the first few hours of daylight. That's clear.
Right. That's okay. That's kind of interesting because, I mean, one of the things is what we should do is reduce blue light in the evening to help you sleep. And so
Definitely.
use red light at
Definitely.
night. But
Yeah,
okay,
definitely. Yeah.
do that as well. Okay. But in terms of improving the glucose and the mitochondria in the morning. Okay. Last kind of topic. So where do you see the future of red light? What are the kind of the most exciting things that you're working on, and what will be next?
We all have different things that grab us and interest us. I have a small cohort of children who have mitochondrial disease. They are relatively immobile. They're in a poor shape. Many of those children will not make it to 30. We took a punt, and well, people contacted me and said, I've got a child with mitochondrial disease. What will happen to red light? And I said, I don't have ethical permission for this. I cannot do, but if I were in your situation, I might do A, B, and C. The results were stunning. And the results we've got in a small clinical trial now are also stunning. And I will be totally openly honest about this. When I saw the results from the first child, I went to the toilet and cried. That was the apex of my career. And that child and the others are still doing well. They will have to use light for most of their lives. When they don't use light, they drift back a bit, but they're mobile as well because the mitochondria are talking to one another. These kids, the patients come back and say, well, yeah, the eyes are opening better, which is what I was looking for as my metric. And I said, anything else happening? Oh, yeah. My child's walking to school now and they couldn't do that before. And I went, I'm totally shocked. The clinicians were totally shocked. So if you talk about me and what I want, I just want to see these kids walking to school. But I also want, as a key target, I want us to age better. And so far, there are a load of metrics I've not explored. I really want to do that proper study on cognitive ability in people, but I've absolutely no idea how to get that funded. There is so much we can do. I want to take a whole load of aged people and stand them on one leg. You stand on one leg with your eyes closed. The older you get, the quicker you fall over. Let's do all these simple metrics with red light. But all of these cost money. And so I'm very, very selective about where we throw our energy and efforts. Throwing some energy into working with engineers so that when you walk along, we have optical technologies that might be able to say, when you walk past me, okay, I give you a burst of red light, it might be able to control your blood sugars, but I can also view in real time what your mitochondria are doing, right? So I can choose people who are vulnerable. Then we can treat those vulnerable people, and we have to do it cheaply, and we have to make people-centered. You don't want to go and use a big machine. You want something that you buy that is almost disposable, that you put in your pocket and is yours. You're in control of what's going on. I think we'll get there, but we won't get as far as I want by the time I die. And that's a little bit annoying.
Yeah. Okay. No, that is wonderful, especially the thing with the children. I just actually a question for getting red light into the brain. Do you think like a nasal device would be more effective?
I know a key player who I respect beyond belief, who actually does exactly that. He uses a nasal device. There's so much stuff we need to talk about and discuss. I've never explored it great with him, but he did say it and I killed myself laughing. But then I said, yeah, that makes perfect sense, because it's going to penetrate into the cranial cavity really well, and it's going to penetrate deep in the brain, deep in the thalamus. Yeah, I think that's fine. There's a lot we don't know. You know, again, we need a project where funded, where we take people, we put the light up their nose, we put the light in their ears, we put the light on the top of their head in a cap. What's best? What's best? What's cheapest? What delivers?
Right.
That's the important point. But yes, it may sound a bit bizarre sticking a thing up your nose and your front of your face goes red. As far as I know, that works.
Okay, so was there anything else? Is there anything else that you'd like to share that I haven't asked?
I think I've said all the things I want. I want people to be motivated to use these devices in aging. I want people to use their leverage to try and get this research funded, because it will make a difference. And I say this for a lot of podcasts, and it cripples me on a Sunday afternoon. Very often, if you've got a question and you email me, I will try my absolute best to get around to answering it.
Okay.
So, yeah.
Thank you.
You're welcome.
Okay, Dr. Jeffery, thank you so much for joining us today. It was great talking to you. Really enjoyed it. And yeah, I mean, good luck with your research.
Great. I enjoyed it as well. Get the word out there.
Will do.